Prepare the Agar plate. You can label the bottom of the Agar plate with the necessary information, including date and sample name. Ensure the Agar plate is sterile and properly labeled. Sterilize the inoculating loop. Hold the loop with the wire loop end facing down. Pass the loop through the flame until it becomes red hot. Make sure to keep the loop in the flame for a few seconds to ensure complete sterilization. Then allow the loop to cool down for a moment, but do not touch the wire loop with anything non-sterile. Inoculate the plate. Lift the lid of the Agar plate slightly with your non-dominant hand while holding the sterilized loop with your dominant hand. Touch the loop to the culture to collect a small amount of the sample. Quickly streak the inoculum onto the agar surface in a zigzag pattern. Ensure that this streaking starts from one of the ends of the plate and ends at the other end. Sterilize the loop again. After the first streak, sterilize the loop as mentioned in step 2. This sterilization step is crucial to prevent contamination between the streaks. Streak for isolation. With the sterilized loop, drag a small portion of the streaked bacteria from the first section into the second section. Again, streak the inoculum in a zigzag pattern over the second section of the agar. Repeat sterilization and streaking. Repeat the sterilization and streaking process from step 4, then proceed to the third section of the plate and finally to the fourth section. Incubation. Incubate the streaked agar plate for the appropriate temperature for the specified time, typically 24 to 48 hours. By following these steps, you can effectively use the streak method with an aseptic technique to isolate and cultivate any bacteria of interest while minimizing the risk of contamination.